This is RTP. This is RTP180.
Cool. Hi. So I'm Yevgeny Brudno. Thanks for the invitation. So I started my lab a couple of years ago, focused on drug delivery systems, and specifically the thing that really motivates me and I think motivates a lot of the people in my lab is thinking about how to get drugs more efficiently to tumors. And so I have this cutesy title about how to get drugs to a tumor, the whole tumor, but the major thing being nothing but the tumor. And so the thing that motivates us is how inefficient and quite frankly sucky current chemo and immunotherapies are. So taking a step away from efficacy, where I think we still have a lot of work to do, just the sheer number of side effects and the sheer number of issues related to cancer therapy really motivates us to do better. So issues with IV infusions or even oral doses of chemotherapies and now the newer immunotherapies, the drug goes everywhere. There's widespread diffusion of drugs throughout both cancerous and healthy tissues. Because it goes everywhere, you need to give excessively high doses of it. So you pretty much give as much drug as you can, as long as you don't kill the patient. Hopefully you kill the tumor, right? And then, since it goes everywhere, and you're giving high doses, and the drug dissipates and clears from the body really quickly, you pretty much have to repeat the doses. So I think anyone that's been through chemo or has had relatives go through chemo, you know the sort of IV infusion system of going every couple of weeks getting these IV infusions. For many decades, engineers, bioengineers like myself, have been working on various local delivery systems, so systems, something you could implant or put in the body to locally release these drugs. But one thing that's been missing from all of these systems is that they've been single use. You do a surgery, you remove a brain tumor, you put in a little wafer. So these right here are gliadel wafers. This is a clinically approved system that you put in the cavity left over after a tumor resection. It releases some drugs, but you're not going to repeat that. So what we think about a lot in my lab is how do we make refillable drug delivery devices that release chemotherapeutics or immunotherapeutics in a local way over a long, sustained period of time. And what we've hit upon are systems that capture inactive refills from the blood, concentrate those refills at a tumor site, and then go away. So they're cleared, the patients pee them out, the refills, except for the stuff that's stuck at the tumor, which allows for slow presentation of our drugs at the tumor site. So there's a lot of chemistry here, there's a lot of biomaterials, and in 5 minutes I'm not going to be able to go through it, so I'll just give you a quick schematic of what we're doing. We take drugs and we make them inactive. We essentially break them to create prodrugs that look like that. And at the tumor sites, we actually label our tumors with some sort of catch motif, something that's going to catch our non-toxic refill. So what's circulating in the blood is our non-toxic, we call these molecules prodrugs. That's what's circulating. They don't have any effect, and they also don't have any systemic toxicity. But what they can do is catch at the tumor. And over a long period of time, a sustained period of time, there's a special chemistry that happens that cleaves it off of the catch motif and at the same time activates it to present drugs just at the tumor site and never in the heart or the liver or your head where you might lose your hair or somewhere else. So these have some, I think, benefits, and we're still very much in development for these systems, but we think that this prodrug, and we've shown this, protects you from the systemic side effects of the chemotherapy. These gels, or now we have new formulations, catch these systems, and so it allows you to concentrate a large amount of drug in a small space right where the drug is needed. The uncaptured refills are just cleared from your body, and the linkers, the kinds of chemistries that we create in these systems, allow for slow release, so we can have a release over a period of a week or over a period of a month. So this is how it works. This is just 2 little gels. One is a control gel that doesn't have our special chemistry. One is a gel that does. If anyone in the room likes and knows a little bit about click chemistry, these are the molecules that we use. And so you can see that in the control gel, our drug surrogate over here is just in the tube. Whereas in our click gel, the drug surrogate has been completely soaked into our gel. And we can now do this. We can start in mice, and then we can go into tumors. So these are mice that have our special chemistry, or control mice that don't have our special chemistry. The signal is coming from a molecule that's IV infused into these mice. But after 24 hours, it's only present where we've introduced our special chemistry. And these are tumors. These are pancreatic tumors. And you can see that pancreatic tumors labeled with our chemistry catch our drugs, whereas tumors that don't have the special chemistry don't catch our drugs. This is a 3D imaging of a tumor that's starting for us to start to understand where these drugs are going, how they're getting there, what is their distribution, are there places we're missing, are there places that we're hitting particularly efficiently. So we can start to understand within a tumor, where are we localizing our drugs and what effect is that going to have. So this is a brain, so we can do a lot of these similar kinds of things in the brain. This is showing very specific. The red is the whole brain, and the green is our, what we call the depot, the place where we're localizing our drug. So if you want to get a drug very specifically to just one part of the brain, we would argue this is one way that you could start thinking about doing this. I won't go too much with this. We have some proof of concept data. These are tumor growth curves. We have a bunch of controls here. The big takeaway is our systems work as well as giving IV bolus chemotherapy, but without any of the side effects. So when we give chemo, we see loss of hair, lethargy, dehydration, weight loss. We see some toxicity studies. We see no side effects with our prodrug systems. And we're taking it in a lot of different directions. We're working on brain cancer. We're looking at lower limb ischemia. We're looking at infection, local infections in bone. There's been some studies with rats on mimicking side effects. So this is my lab. This is what we will work on, and this is what we're excited about. And I look forward to talking to you after the presentations.